Hello, and welcome to She Leads Biotech, the podcast that celebrates the achievements of women in the bioscience space. I'm your host, Sharron Cox, CEO and Managing Director of CNA International, a boutique search firm based in New York. Each week, I have the privilege of learning from incredible leaders as they share their stories. Together, we celebrate their triumphs, explore their challenges, and uncover their vision for the future of bioscience. Our conversations also provide invaluable insights into how women can unlock their unique superpowers and succeed on their own terms. So, whether you're just starting your journey in bioscience, contemplating your next career move, or simply passionate about science and innovation, this podcast is here to empower, inspire, and guide you. From a childhood fascination with science to leading groundbreaking cancer diagnostics, my guest today has always been driven by discovery. Choosing impact over convention, she built her career at the intersection of genetics, oncology, and cutting-edge diagnostics. Whether at a global pharma company or a nimble biotech startup, she's thrived by building high-performing teams and pioneering new approaches to personalized medicine. Now, at the forefront of cancer detection, she's shaping the future of diagnostics with relentless curiosity and leadership. So please welcome my guest today, Chief Science Officer at Droplet Biosciences, Wendy Winckler. Hey, Wendy, good morning.
Hi, Sharron. Good to see you.
And a happy Friday to you.
You as well. We made it.
Another weekend is almost here. Thank the Lord. Well, listen, it's great of you to join us. Thank you so much. I know how busy you are. You're in Massachusetts. You're in Cambridge, right?
I am, yeah. My work is based in Cambridge.
So we're in the same time zone. So that's good. How did it start for you as your career? I've got to be honest, you're my first chief scientific officer. So I'm really intrigued. Did you pick the field? Did it pick you? How did it all start for you?
So science, I think, picked me. I've pretty much always known that I was going to be a scientist of some kind. I mean, as a preschooler, I was obsessed with astronomy and space science. And really liked lots of different aspects of science. I discovered genetics, which is what I ended up doing my PhD work and largely everything I've done since in middle school when I took a class at the University of California in Irvine that was a summer program on genetics and really a deep dive. And I just fell in love with it and I spent a lot of time thinking about it, like outside of school and everything, majored in it in college and have been doing it ever since.
Wow. Okay. So you got a you got a PhD in genetics, or was it in? Yeah. Okay. And so your first foray into work from that was what? Where did you go?
So when I finished my PhD, I was trying to decide what to do next. And I had the good fortune of doing my graduate work with David Altshuler, who was one of the founding members of the Broad Institute, which, and previous that had been the Whitehead Genome Center, one of the groups that was doing the human genome sequence. And so while I had, more of a traditional graduate dissertation project, I got to be in an environment where I was seeing all of these giant scientific consortium projects. And I realized when I was finishing up that I didn't want to do the traditional academic path. I didn't want to be the principal investigator of my own little lab. I really wanted to be a little part of something big that was going to change science, medicine, for everyone. And so I decided to stay on at the Broad as a research scientist. And I joined the cancer group there, which was just getting involved in the very earliest cancer genome studies. And so that really shaped from there. I think I realized that I love science. I think I've done a lot of great scientific work, but maybe my biggest differentiator is that I'm really good at kind of driving organization and delivering results, bringing together cross-functional teams to help guide that. And so that really shaped my career being part of these genome projects, and have done that since.
And in the early days of your career, was this something that many women did? Were you kind of part of a big group of women that were involved in this, or was it quite male focused?
Actually, that was probably one of the things that shaped the direction I took, is that there were a number of women staff scientists at the Broad, at the time that I was, finishing my PhD. And I was really attracted to the fact that these women had, powerful roles, but they were always these cross-functional roles where they were working on bigger, broader projects. And I also liked the fact that they seemed to have pretty good work-life balances.
Right.
Compared to, say, the academic professors that I had seen, kind of going through training. And so while, I was, 26, 27 and wasn't really ready to pull that trigger yet, it was in my mind that was something I was going to want to do down the road. And so seeing those role models was important for me in picking that path.
Okay. So just one of the things I love about doing this podcast is the schooling that I get in various aspects of science. Talk to me about your work in genetics and what it means and what the sorts of things were that you were working on and are still working on and evolving. What is that?
So at its most basic level, what we're doing in genetics is looking for a change in DNA that's causing, a different phenotype. In many cases, it's causing a disease. So when I started my graduate work, I was looking for what they call common variations. So these are changes that exist throughout the population, but some of them make you more likely to get, in my case, what I was studying, type 2 diabetes. So looking for the things that weren't single genes that drove a disease, but instead the collection of the many different changes that we all have that, add up to your risk of whether you're going to have some of these really common diseases like heart disease or diabetes, neurodegenerative diseases, things like that. And so I did that for a while, and then I got really into this cancer genetics, and that was particularly interesting at that time because you're looking for your own cell will have a spontaneous mutation as an adult. And that change allows, and usually it's several changes that happen in succession, allows that cell to grow out of control, causing this cancer. But what was attractive in that is if we could find what that change was, we needed to then just turn that cell off to kill that cell. And we could, try, hopefully from knowing the genetics, be able to develop rational therapeutics that would lead to that. And so there was this really exciting couple of decades now of work where genetic discoveries were going hand in hand with therapeutic development. And a patient would then go through some of these genetic diagnostics. And if they had something that therapy was available for, they'd get a very personalized medicine treatment given to them. So that's been a lot of what we're doing. What I'm doing right now is continuing to work in cancer diagnostics. So instead of choosing particular therapies, using genetic tools to understand if disease is left behind after we've tried to treat these patients.
Oh, wow. Okay. And we can talk a little bit more about where you are and what you're doing at the moment. But what was your first foray into industry from where did you go? Where was your first role within industry?
So I was at the Broad for a number of years, but then was recruited to join Novartis, a large Swiss biopharma, by someone I had worked with earlier in my cancer genome studies, Bill Sellers, who is now running oncology research at Novartis. And I was recruited to start up a totally new department there called Next Generation Diagnostics. And the goal was to bring the types of exciting technologies we were using in the research world to change how biomarkers and diagnostics were being run in oncology clinical trials. And so it was an amazing opportunity. I got to build a 40-person team from scratch to develop an entirely new group and a new approach to how we were doing biomarkers in pharma. And we were incredibly successful with that. So we were the first pharma to launch liquid biopsy as something that was incorporated into all of our clinical trials, really from the earliest, first in human types of studies all the way through registration studies. And whatever we were really interested in. So while I was there, immuno-oncology became a really big theme in cancer, but we didn't have any great tools or biomarkers to understand who was going to respond, why were they responding, can we see, what this new drug we're developing is doing in that patient. And so some of the genomic tools that my team had been developing, things like spatial sequencing, so like being able to see it on a slide, RNA sequencing, so understanding not just what the genetic changes are, but how are those being expressed and made into proteins. So we were able to employ that to try to get a better understanding of how immuno-oncology was working, because it was a totally new area for all of us.
And how did you, as a manager of these teams of people, what's your leadership style?
Interesting question. I like to bring in the best people that I can find. And in that particular group, a lot of that was not necessarily the most experienced people, but I looked for people who had a really, great talent in a particular area and then a passion for being able to drive that. I also created a really multidisciplinary team. So a lot of it we haven't talked about yet, but in genetics and the genomics revolution we've seen, there's a laboratory component, sure, but equally important here is the computer science aspect. So these data sizes are so huge, we need to have really sophisticated computer and data science experts to be able to understand the data, to mine it, to make the discoveries. And so the team I created was half what we call wet lab and half of this bioinformatics data science. And so a lot of what I did as a manager was find great people, but then coordinate them. So I set vision, I aligned, and worked really hard to make sure that each of those people, I could take obstacles out of their way and make sure that they were working together to achieve great things.
And was that a mix of men and women team? What was the team dynamics?
Yeah, it was a pretty diverse team. My leadership team was, I think at the beginning, it was equally men and women. Actually, at the time I left, we'd done some restructuring, people moved on. I ended up with 4 women, which was a really interesting group, my power team there towards the end. But yeah, the dynamics were a lot of fun. And it was great to be able we had so many women leaders, PhDs, women data scientists, which is not the most common, that were part of that group and have gone on to many of them moved on to other roles, but are still doing fantastic things in science.
Okay. And so I believe when you left Novartis, you joined almost a startup biotech. Oh, yeah. What was that transition like?
That was really different. So I went from, a large global pharma company to what it was at the time. I was employee 12 at a Series A stage startup. So that was a big shift, and then additionally, I moved from cancer and genomics into liver disease and protein-based work. So for me, it was almost a complete reset of everything I'd done, which was both challenging. And for me, it was, I think, the biggest growth period of my career, just because I really, everything was done totally differently. But you asked what was different about going from pharma to startup. I think the biggest thing you kind of notice at first is just the lack of bureaucracy. So when we were able to go so much faster, it's, instead of, okay, we need to make a decision on this important, our program, at Pharma, it would be like, okay, we'll put that on the agenda for our global decision board in 2 months. And then, and then we will spend weeks preparing a very formal slide deck, and we will go in and we'll have practice talks and all these things, and we'll make a decision. And in a startup, like an hour after the problem arises, you grab the 2 or 3 people who need to be there and you go to the whiteboard and you hash out exactly how we're going to deal with that. So that was really different. At the beginning, it was very freeing. And then sometimes it was very exhausting because the pace was so fast all the time. Yep. The other thing, though, that I think was hard is in a big company, you have so many resources and talented people. There's an expert in everything. When I went to Glimpse, which was the startup I did first, I was the chief scientific officer. I was running research, but I was also running clinical, which included regulatory, like manufacturing, all the different things that we were for our drug product. A lot of this I had no previous experience in, so I was learning constantly, and there was just no one you could call who absolutely knew it. You had to go out and find an expert or learn it yourself or make these decisions. And that was challenging.
Yeah, it's kind of like, oh, I'll put my manufacturing hat on today or for this hour, and then I'll put something clinical on and work my way through that. How did you find that? I mean, obviously you're still in relatively small setups. So did you initially want to run back to big pharma, or did you embrace that challenge and enjoy it?
Oh, I really enjoyed it, actually. It was being able to have a small team that's incredibly focused on that one program. Because in a very early startup, that's usually all that there is budget for, and everything is relying on that.
Yep.
At a big company, there's a large portfolio, and everyone's working on a lot of different things. Everyone is in the same boat and rowing in the same direction, at least in a functional startup. And so that was the ability to do that, to make progress. The pride of ownership, I think we felt too, was something like I just not experienced in the big pharma setting.
Excellent. So where you are at the moment, Droplet
Yes.
Biosciences, right? Talk to me about the work that they do and what you do there.
Sure. So our company is working to get an earlier read on whether a patient has residual cancer after they go through surgery to remove that cancer. And we're doing that, though, not by working through a blood test, but instead by taking the lymphatic fluid right from the site of surgery. So this is a fluid that no one's really ever worked with before. But because it's coming directly from where that tumor bed was, we see that there is an increased concentration of tumor-derived analytes when there is residual disease remaining. Whether that disease is hiding in a lymph node that wasn't removed, or what we call a micrometastasis, and it just doesn't show up visually. Those things are shedding into this fluid. And when we tap into that, we're able to get a much more accurate and earlier assessment of whether that patient still has disease. And that's important because it's allowing the patient and their care team to make better decisions on the next step, which is adjuvant therapy.
Right. And again, I know nothing about this, so I'm just learning it as I go along. By doing that, if your diagnostics are saying there's still some stuff there, whereas previously they may have said, we're just going to do this operation and cut the cancer out, would be we need to do chemo or we need to do something else to make sure we get everything. Is that right?
Exactly. So right now, the guidelines that clinicians follow after surgery are based on pathology. So they're based on what was taken out of the body. So the tumor or the lymph nodes that might have been excised, but they really can't see what was left behind. And those guidelines will say, okay, you know what? The lymph nodes were clear, just watch this patient. Or we see that there's, literally still positive surgical margins, meaning that we know cancer was left in the body. Let's treat that patient maximally and give them radiation and chemotherapy. But there's this intermediate group of patients that have maybe one or two slightly higher risk factors, but not the highest risk ones. And that's where we've seen the droplet may be really effective based on our retrospective studies. Is that we can really stratify those patients, those ones who go on to have their disease come back within the first year versus those patients who, really have no evidence of disease for years after the surgery. And so for those patients, because the head and neck cancer that we've started in is really aggressive, that can be additional information that would say, instead of just giving that patient radiotherapy, let's go ahead and add on a systemic treatment like chemo. And hopefully that will lead to more personalized treatments, but also better outcomes.
It's difficult with trials for head and neck cancer, isn't it? Because it's actually quite rare, but it's devastating for those that do get it and how the treatment proceeds. So I would imagine the diagnostic area that you're able to do can really cut back on the ongoing work that needs to be done. Does that make sense?
So it is the sixth most common cancer in the US.
Really?
Yeah, it is.
Okay.
It's pretty common. You don't hear about it as much as you hear about lung cancer or breast cancer, but it is, because this includes all of the cancers of the mouth, throat, in that region. But it is one of the cancers that doesn't have a lot of diagnostics out there. Things like lung cancer have screening protocols that are put in place to try to catch them earlier. We all know about mammography for breast cancer, for example, to try to catch these. But we don't have the same early screening, so things can be diagnosed, later, typically when the patient is having symptoms. But then, because we talked much earlier in this about the idea of a genetic driver, and then there's a targeted therapy that can be selected, head and neck cancer doesn't have a lot of those. And so there haven't been very many diagnostics developed in that space.
Okay.
And so we are hoping that our test is going to be able to make an important impact for this disease.
Excellent. Okay. I ask about head and neck cancer because I've signed up for their move-a-thon that happens, which anybody who knows me knows I really don't move that much at all. Should perhaps do so, but I don't. So I'm trying to kind of digest as much as I can about the work. It's the Neck and Head Cancer Alliance and the work that they're doing in order to be able to sell, getting more sponsors for this little move-a-thon thing that I'm going to do. So it's interesting to talk to somebody whose company is working in this area. And I obviously wish you every success in doing that. Something else that I've seen that you've been involved in is blood pack.
Yeah, we just recently joined that.
Yeah, what's that all about?
So Blood Pack is an academic and industry consortium of people who really want liquid biopsy to become an important tool throughout cancer. And so they are helping to set the policies, best practices for those types of things. And they do that, so there's people involved from government, from FDA, from NIH, the biggest, pharma companies, the big diagnostic companies, but also some smaller companies like the startup I'm at, Droplet. And so, working together to really understand the things that we all have to solve, like, what are the right tubes to collect this fluid in and to keep this material stable? How should we design validation studies? So those things that have to happen, they're very technical, but also some of the things that are really important for diagnostics to succeed, like how do we get reimbursement? How do we design clinical trials to best get through FDA and get approval for these types of tests? And so people are all bringing and sharing their expertise. There's scientists, there's business folks, there's clinicians, regulatory experts, all coming in and driving that. And so the group that I am now co-chairing is the newest working group at BloodPack, and it's called the Multiomics and AI Working Group. And so most of what's been done in liquid biopsy in cancer to date has been DNA-based, but there's a lot of interest in adding in other types of analytes, things like proteins, metabolites, or other things that might give us additional information and maybe make our tests more sensitive, more accurate. But how to go about doing that, I think, is a real challenge, and one that all of those different groups I just talked about are all kind of facing. So both, how do we generate this data appropriately in a way that's going to give us really reproducible results? But then how do we use some of these new AI tools that are coming out that are so exciting and powerful, but if you don't use them correctly, if you don't design the studies right, you're going to get overfit models and you won't have a test that's reproducible when you take it out into the wild. So we got a really multidisciplinary group of scientists that have come together to start planning these. And our first step is really to just start to define the lexicon, the vocabulary. Let's all say that we're meaning the same thing. And that's something that we'll put together a publication on and put that out there to help guide the whole community.
Excellent. Well, I wish you all the success in the world with that one. What does the future look like for you? What does Wendy Winckler have in her future?
Well, I'm very excited about the current product that we're about to launch at Droplet. So right now we're building a CLIA lab for the first time in our group, and we're taking our first test, which is head and neck, into that so it will actually be available for clinicians to order as a test later this summer. So that's, for me, like when I joined the company, it was a great idea, but there was no data behind
Right.
whether this was going to work in HPV negative head and neck cancer. And so in 2.5 years to bring that forward into something that's going to actually be used to inform patient care is pretty exciting. And then beyond that, getting this to work in other indications. So head and neck is our first place. It was a great place to start, but we're also working now in bladder cancer, in lung cancer, and beyond that, other types of cancers will follow, and to see if we can bring the same type of approach there. So then beyond that, where will I go once we do that?
World domination?
I'm kind of open-minded to a couple of different paths, and I'm not sure where that'll end up. I've had a number of people tell me I should think about the CEO route. So for my Novartis experience, I do love to build. I love to establish teams. I love to grow concepts and set the vision and that. And I think that's a really important part of CEO roles. There's also a lot of it, which is, fundraising and managing more on the business side, which would take me away from the science, which was my first great passion. So I'm exploring, I have to think about that one. That is maybe the more conventional, like what you do after CSO path.
Right.
I'm not opposed to going back to the big company route either, kind of taking what I've learned from being nimble and startup, learning to go fast, learning to do without, like, especially in the current fundraising market, like, how can we design this study to be equally effective, but maybe cost less? How could we, how can we do this with the smallest, most able team we can? I think those things will suit me well back in a big environment. And so, I don't know, I think what I've never done, I've been in big pharma, I've been in small diagnostic startups. I've never done big diagnostics. And so, one of these companies that has, been around for years and has several established products, and then, is also doing some research and developing the next generation of things. I think that could be a really interesting place to apply both of those skill sets that I've learned.
Do you think you'd find, because I don't know that the bureaucracy in big companies will ever go away. And if you've been in smaller environments where everything has been so fast, what do you see that learning curve being of actually going back into something big and maybe trying to speed up a process that's been kind of chugging along or however it's been doing for whenever? What sort of challenge do you see that being?
So I think there will be some frustrations with some of the, I guess, bureaucratic procedures that are put in place or the systems that you have to use. But I think that there will also be a rush of, like, once again, I have all these peers and colleagues who know so much that I can leverage, I can lean on, I can partner with. Because, being in a startup can be pretty lonely sometimes too, right? When it's a small group, you got to run your part, and there's not always somebody that you can run to and share the experience with or get advice from. And I have been really fortunate to be part of a CSO small group, sort of peer coaching and mentoring type group, which has been incredible. But I think in larger companies, you have some more of that just baked in.
Yeah, you don't have to work quite so hard to move through with that. So what was that journey like for you to CSR? I mean, we've spoken in this conversation and it almost appears that, I did this and then became a CSO, but I'm pretty sure it wasn't as straightforward as that. What was that path like, and what challenges did you find on your way up into leadership?
So my big jump into leadership happened way back in the Broad years, where we talked about working on the early cancer genome project. I was like the lead biologist for the Cancer Genome Atlas, which was NIH's, original signature cancer genome project. And from there, I had an opportunity to take over a 40-person team called the Genetic Analysis Platform that had been already established, was running thousands, tens of thousands actually, of patient samples per year. A fully baked team with an ongoing portfolio of projects. I had never managed people before. never really had to manage a budget. And all of a sudden I was dropped into this role, which was a real trial by fire opportunity. It was definitely probably the scariest leap I've made, but I loved that group. I found that I really liked the operations. I found I loved people management in particular. And I learned a ton of those things. So that for me was the big jump. The Novartis jump, which was the next one, was, a similar sized team, a lot of that, but that brought me back into the science a little bit more as well as the challenge of now navigating a whole pharmaceutical company. So the CSO jump, like when I left Novartis to go to Glimpse, I think I was just really ready for just a new challenge, a new way to try something. My team, NGDX at Novartis, was now 7 years established and doing great, but I missed the build phase. I missed kind of that rush of the early. And so that's what called to me about the startup world was doing that building. Kind of unique feeling you get when you're a less than 20 person company. Like I said, all kind of rowing after the same thing, and you all get to know each other incredibly well. There's a family-like vibe when you're that small.
Days of a relationship, isn't it? When you're all just,
I guess,
and then you're in love, and it's all lovely, and then in some instances it all goes. Then okay, I've got to move on from this now. We've done our time together. It's been fabulous. Please don't think it hasn't, but time to move on.
Yeah, I think that happens too. But I liked that in companies, I think, as well as personal relationships. I did, that was the driver there, and I think it's true. You do have that very much in the startup world, and I got to enjoy that again at Droplet.
Cool. So, what would your advice be to somebody who's maybe doing a PhD at the moment, who's really not sure what their future looks like? They think that they're very keen on the science side, but not sure where to take it. What would your advice be to somebody like that?
So I think be open to lots of different types of opportunities. So when you're doing graduate training, you're necessarily mostly exposed to the traditional academic path, like postdocing or, being a faculty member in an academic lab. But there are so many other opportunities for what PhDs can do that it's worth exploring during that period. It's worth networking and really building relationships in a lot of different directions, whether that is something like going into industry as still very much on the scientist's side, but in a different environment. But there's, you could go into investing or other types of more advising roles where you use that science background, but you're clearly doing a very different kind of work. To things like project management and, regulatory specialists and all. So I think keeping an open mind, asking a lot of questions, watching a lot of, and seeing what resonates. But the other advice is not just for the new grads, but, as you go through, just because you started on one path doesn't mean you have to stay there. There's lots of opportunities to sort of turn corners and try something new. And that for me has been a big part of the learning opportunities. I've been in so many different environments now. I spent a lot of time in academia, but I also was in big pharma. I've been in small biotech. And that, for example, in my current role, it makes it a lot easier for me to do business development types of discussions with big companies because I'm familiar with that culture. I'm familiar with how they evaluate what they look for.
Right. Which also prepares you wonderfully should you choose to look at the CEO route further down in your career. They're all preps ready for the next stage, right?
Right, I agree. And I think I've gone back to leverage a lot of different experiences. Sometimes they were things that I didn't find all that fun or helpful at the time, but it was, it's incredibly useful to have that experience and be able to rely on that. The other thing is I've gotten to know and work with so many different people and so many different kinds of specializations that it's been amazing to be able to call people up to get advice and sometimes to hire them again into different companies and to partner. For example, we were talking about blood pack, but so it's one of the other fun things about this new group that I'm chairing is one of my co-chairs was the head of my laboratory that I hired at Novartis. So for us, we found another way to get to work together and to continue that relationship.
Excellent. Well, listen, it's been an absolute pleasure speaking to you. I know you've got a busy day ahead, so I will thank you for your time. And I can't wait to see the success that you continue to have at Droplet and the success that Droplet has in moving forward with these diagnostics that can only be wonderful for, patients as they go through these things.
Thank you so much, Sharron. I really enjoyed talking with you today.
Yep, absolute pleasure. And we'll speak again.
Sounds good.
Take good care.
You too. Bye.
Bye. That wraps up today's episode of She Leads Biotech. I hope you enjoyed this conversation and found inspiration in the stories shared. Remember, the success of every woman can inspire another, so don't hesitate to share yours. She Leads Biotech is brought to you by CNA International USA, a woman-owned boutique search firm specializing in sourcing talent for small and emerging bioscience companies across the US, from our headquarters in New York. For more information about our services, feel free to reach out at contact@cnaint.com. Thanks for tuning in. Be sure to subscribe so you don't miss future episodes. And until next time, stay bold, stay brilliant, and continue pioneering progress for women in bioscience.